Central Dogma Molecular Biology by Matteo Cagnucci. Okay, so today I'm gonna explain the three important processes of Central Dogma. Okay, so let's start. So Central Dogma starts with the process of DNA replication. And DNA replication is a process which a double-stranded DNA molecule is copied to produce two identical DNA molecules. DNA replication is one of the most basic processes that occurs within a cell, normally happening during cell division, so that each new cell has a complete set of DNA. In this way, for this to be done successfully, copies of genetic information must be made before the cell division process is carried out. Okay, so there are three important steps that have to happen to complete this process good. Okay, the first one is inside. DNA allocates inside to DNA strands by breaking the hydrogen bonds. The second one is base pairing, that occurs when free-linking DNA polymerizes nucleotides with leading strand nucleotides. And the third one, nucleotide union. It's the end of the process and tries to make the ligase bonds unite the nucleotides. Okay, so let's continue with transcription. That is the second process of Dogma. What is transcription? Transcription is a process which DNA is copied to make a complementary strand of RNA. The RNA is then translated to make proteins. The major steps in transcription are initiation, its promoter removal, elongation, and termination. So in this way you end up creating temporary copy of a RNA. Okay? And here in transcription there are four important steps that are: first, enzyme. An enzyme decomposes DNA, that is DNA helicase. The second one is the free RNA nucleotides paired with DNA nucleotides. And the third one, when base pairing is done and the new mRNA is broken. And finally, the fourth one is when the mRNA leaves the nucleus and enters the cytoplasm. Okay, so let's continue with translation. Translation is the process which cell produce proteins using the genetic information carried in messenger RNA, summing up in conversion of RNA proteins. Okay, and here are four important steps of translation. The first one is mRNA attaches to the ribosomes. The second one is the tRNA molecule with the correct anticodon attached to mRNA codon. And the third one is the mRNA slides through the ribosomes to next codon and another tRNA comes and attaches to mRNA. And finally, the fourth one is the first tRNA releases into amino acid to attach the second amino acid on using peptide bonds. And finally, the last question of translation is: how is this translation into a protein? And as we know, proteins are formed by the union of amino acids with peptide bond of long chains called polypeptides. Therefore, for translation to take place, the genetic code needs to be read 3 letters at the same time that each codon that belongs to a particular amino acid becomes 3 long mRNA bases. Okay? So mates, this was my presentation about Central Dogma of Molecular Biology. So this was all, and thank you for your attention. Bye.